Hello and welcome. This video is going to cover some of the common methods and techniques used to study gene regulation and expression. Let's start by reviewing the 5 levels of gene regulation. First, there's genome level regulation, followed by transcription. Third is RNA processing and export. Then there's translation regulation, and finally, post-translational modifications. There are many techniques that can be used to study gene regulation at each of these levels, but we're going to look at some highlights that will be useful in class or that you might come across in the primary literature. Let's start off with the genome. How do we study if the DNA that makes up a gene is accessible to the transcriptional machinery? How DNA is packaged in a cell is vital to gene regulation because it controls whether or not the DNA can be transcribed. DNA is wrapped around a histone octamer called the nucleosome. The short stretches of DNA between nucleosomes are known as linker DNA. As drawn below, the DNA is relatively open or accessible, meaning that transcription factors can bind, nucleosomes can be repositioned, and transcriptional machinery, like RNA polymerase, can access the DNA. However, nucleosomes can also be tightly bundled with the help of proteins such as histone 1 or H1. A good analogy for DNA packaging is a row of movable library bookshelves. When squished together, like with condensed nucleosomes, they save space. But if you want to access a book, or say a particular DNA sequence, then they have to be shifted. Although generally stable, like bookshelves, DNA packaging can shift between condensed and open configurations in a process called chromatin remodeling. How can you tell if a gene you're studying is in an open or closed configuration? The answer is with DNase, a protein that cuts exposed DNA right in those linker sequences. The reason the DNA sensitivity assay works is because when parts of the genome are under active transcription, they are unbundled from condensed chromatin, making them accessible to DNase. In this assay, you add varying concentrations of DNase to the samples with intact chromatin. You then extract DNA from the sample, releasing it from any proteins. The samples are then digested with restriction enzymes to generate fragments of similarly sized DNA. Finally, the samples are run on a gel and a southern blot is used to detect the presence of DNA fragments. A southern blot uses labeled DNA probes that bind to a particular sequence of DNA. As a scientist, you design the probes to interact with whatever DNA sequence you're interested in. If your gene of interest is in an open configuration, as the concentration of DNase increases, there will be less and less of the fragment of interest because it will have been cut into smaller pieces during the DNA digest. Let's take a look at an example. Suppose there are 2 genes on the same chromosome. One is involved in the cell cycle, while the other is involved in synapse development. You're investigating 2 different cell types, neural stem cells and neurons. What result might you predict if you perform the DNA sensitivity assay? Let's set up the blot. This is the chromatin configuration that you would expect to see for each cell type. Back to the gel, I've indicated DNase concentration above with the triangle. Based on the probes we designed, the genes show up at 2 different sizes. In the stem cell sample, you would expect to see bands of equal intensity across all DNase concentrations for the synaptic genes because these cells don't form synapses. However, since stem cells can undergo mitosis, or cell division, you would expect that the cell cycle gene is sensitive to DNase, and so its corresponding band would decrease as DNase increases. In your sample of neurons, the opposite is true. You would expect the cell cycle bands to remain constant across DNase concentrations because neurons don't divide. However, the synaptic bands this time would decrease, indicating that this gene is actively transcribed in this particular cell type. We just talked about the DNase assay, which can be used to characterize the accessibility of the DNA to the transcriptional machinery. Now, we want to know if there are indeed proteins, such as transcription factors, that can bind to that DNA. This can be studied using EMSA, or electrophoretic mobility shift assay, also known as a gel shift assay. This assay uses a non-denaturing gel to preserve the interactions between DNA and the proteins bound to it, which can then be visualized using labeled DNA probes. Recall that with gel electrophoresis, smaller things move more quickly through the gel. DNA is also negatively charged, so it will be repelled from the negative charge that's applied to the top of the gel. Therefore, all alone, the DNA probe will appear at the bottom. However, when it's bound by a protein, it will move more slowly and appear shifted upwards in the gel. Now, there are a number of different controls and additional experiments that you can run with this type of assay. For example, let's say that you incubated your probe with total cell lysate to confirm that your protein of interest is in fact shifting the probe and not some other protein. You can incubate your sample with an antibody against that protein. This will super shift your band in the gel. What if you're interested in the sequence specificity of the binding interaction? It's possible to determine a consensus binding sequence, or you might want to determine how a mutation affects the binding of that protein to your DNA sequence. For example, you might compare the binding affinity of a wild-type probe with several mutant probes. Here, mutation 2 reduces binding of the probe in comparison to wild-type, but mutant probes 1 and 3 are no different. Let's shift gears. So far, we've discussed assays to test whether DNA is accessible and can be bound by proteins. Now, let's talk about how we can measure protein using a Western blot. The basic procedure involves taking 2 or more samples you plan to compare, lysing the cells, extracting protein, denaturing the samples with heat and a detergent, and running them on a gel. SDS Page is commonly used in Western blots. SDS is a type of detergent, or soap. Think laundry detergent. And PAGE stands for polyacrylamide gel electrophoresis. Polyacrylamide is the material the gel is made of. The same basic principles of electrophoresis that we discussed for the EMSA assay applies to this situation as well. Since proteins are slightly negatively charged, they will move away from the negative current applied at the top of the gel. As proteins move through the gel, they move at different speeds depending on their size and charge. Size can be estimated by running a protein ladder which is made up of proteins of known sizes alongside your sample. Smaller proteins end up near the bottom of the gel. This is what a gel might look like. There are 2 duplicate halves which will be used for the experimental and control antibodies. After running the gel, the samples are then transferred, using electrophoresis again, to a membrane for antibody detection. Most Westerns use a 2-step detection process in which the primary antibody binds the protein of interest and the secondary antibody is used to visualize it. The secondary antibody is usually conjugated to a fluorophore or to an enzyme that will deposit a colored precipitate. Note that this is a very different use of an antibody when compared to an EMSA assay. In a Western blot, we are using antibodies to detect proteins after they've been transferred to the membrane. In an EMSA, we use antibodies to characterize if a protein is bound to DNA and if it migrated differently in the gel. Recall we had duplicate gels so that one could be used for a control protein. Expression of the control protein shouldn't change between the different conditions of our experiment, so it can be used as a loading control. A loading control ensures that the samples had a similar amount of protein to begin with. But if they're a little different, you can use the control protein to normalize the blot with our experimental antibody. This gives us a relative intensity and allows us to compare between samples 1 and 2. Total protein can also be used for this normalization. Our graph of relative intensity shows that there's less of our protein of interest in sample 2. Of course, N of 1 isn't enough replicates, so you'd want to repeat the whole experiment multiple times, starting with fresh samples for independent replicates. Let's take a minute to talk about antibodies, because they can determine if your Western blot works or not. Finding the right antibody can be tricky, but it's vital to a good Western. If there are no good primary antibodies for your protein of interest, there are also other strategies that can be used to measure the level of protein expression indirectly. The yeast lab uses one of these, but I won't get into the details here. Some antibodies are very specific and will only bind to a single protein. But if your antibody binds to part of a common motif, it may recognize that motif in multiple proteins, resulting in off-target binding and making interpretation of your results challenging. Western blots are also used in co-immunoprecipitation, or co-IP, a technique used to study protein-protein interactions. Let's return to NF-kappaB to illustrate this method. Say you're investigating a particular mutation in I-kappaB, the protein that prevents NF-kappaB from entering the nucleus. You hypothesize that this mutation prevents binding of I-kappaB to NF-kappaB, and you decide to test this idea using co-IP. Here, we have wild-type I-kappaB bound to NF-kappaB and a mutated I-kappaB unbound. Now say you know that there's a really good antibody for NF-kappaB, so you decide to use it as your primary. Some protocols call for fixation with formaldehyde, but others rely on the native interactions between proteins. With either, you incubate the samples with a secondary antibody that's conjugated to a magnetic bead. Then, using a magnet, you pull down everything that's connected to the bead out of solution. After washing the samples several times, again using the magnet, you extract protein from the beads and run the samples on a Western blot. We're going to use two different antibodies for the Western blot, but first, for any IP, an important control is the input sample. The input sample is collected and saved before the IP, or before the primary antibody is added. Normalizing to the input allows for comparisons between samples. The input is typically diluted 1 to 10 because there's so much protein in it relative to the IP, since a lot is lost during the procedure. Now that we've run and transferred our samples, what protein should we probe for? Since the primary antibody in the IP was against NF-kappaB, a good place to start is with NF-kappaB. We would expect both samples to contain it, showing that the primary and secondary antibodies worked as expected. But what we're really interested in is probing for I-kappaB. We want to see if the mutant is bound to NF-kappaB. If our hypothesis is correct, we should see I-kappaB in both inputs, since this sample was essentially untreated. And we should also see it in the wild-type sample, but not in the mutant, indicating reduced binding. Changing gears from immune-based assays, let's discuss how we measure gene expression. I'm talking about qPCR or RT-PCR, also known as quantitative PCR or reverse transcription PCR, a method for analyzing the amount of a particular RNA in a sample. The basic procedure here is to take a sample, extract total RNA, and generate cDNA using reverse transcription, hence the name. The C in cDNA stands for complementary, since it will be the complement to the RNA it was made from. A side note on a Wisconsin connection, Howard Temin discovered reverse transcriptase while at UW. You may recognize that name from the Lakeshore Path. Then, PCR, or polymerase chain reaction, is used to amplify specific sequences of DNA with primers. An instrument monitors how quickly the DNA is amplified. Basically, the more DNA in your sample to begin with, and therefore the more mRNA, the faster the sample will be amplified. The general idea is that you can compare relative mRNA expression between samples because you included both a control gene and a control sample in your experimental design. With this relative method, the expression of our genes 1 and 2, which are our genes of interest, are normalized against a gene that shouldn't change under the experimental conditions, such as a housekeeping gene or a basic metabolic gene. Both samples are then normalized to the wild-type sample, which is acting as our control. After analysis, the result will be a bar graph with relative expression on the y-axis and genes 1 and 2 on the x-axis. Since we normalize to the wild-type sample for both our genes of interest, we can use the same scale for both genes, where wild-type is set to 1. Now, what's the expression of genes 1 and 2 in our mutant sample? For gene 1, it looks like there's no difference in the expression between the wild-type and mutant samples, but gene 2 has a lower expression in the mutant sample relative to the wild-type sample. This is another experiment that should have multiple independent samples, so that we can really tell if there is a difference in the expression of gene 2 in the mutant. With qPCR, you can measure the expression of genes one at a time. But if we want to measure the expression of many genes, we need to use a different method, such as a microarray. Microarrays are a way to measure the expression of thousands of genes at once. But since there are approximately 20,000 genes in the human genome, there's no way that a single microarray could ever cover every mRNA. Instead, researchers often use microarrays that focus on genes expressed in specific cell types, pathways, or cell processes, such as in neurons with NF-kappaB signaling or in cell division. How does a microarray work? Basically, you take two different cell populations, let's call them 1 and 2, collect mRNA, and make cDNA via reverse transcription. But you label your cDNA with two different fluorescent dyes. Green and red are pretty common. Then you run your samples on a microarray chip, which has a bunch of different single-stranded DNA sequences printed in discrete locations on the chip. Then you analyze where and to what extent your samples bind on the chip to determine gene expression. A microarray chip is then essentially an array of tiny dots, which is where it gets its name. What does the procedure look like? You combine the cDNAs from both samples, one red and one green, and wash it over the microarray. If the cDNA matches the sequence printed on the chip, then it will bind. Then you scan the chip for the fluorescent signal from both dyes. Locations where the cDNA from sample 1 is bound will appear red. Spots with sample 2 cDNA will be green. Where they both bind will be yellow, and if neither binds, it will be blank. Software packages then allow you to compare the expression profiles between samples and identify larger patterns in gene expression. For example, if you're examining a cancer cell line, you might expect to see lots of changes in the expression of genes that regulate cell division. Finally, I'd like to talk about using gene reporters as a method to measure, monitor, or visualize the transcription of a particular gene. I mentioned this briefly when discussing the yeast Western blot, but let's look at the details a bit more closely. Let's start off with a gene that we'll call gene X. We have the transcriptional start site, the TSS, which is the start of our gene and which is nestled in the initiator sequence. mRNA transcription begins here. The body of the gene is made up of exons, which are the expressed regions of the gene. The first exon contains the 5' untranslated region, or UTR, and the final exon contains the 3' untranslated region. Between the exons are various introns, or introgenic regions. I've drawn them as very short, but they can actually be quite long. Transcription generates pre-mRNA, which is basically an RNA copy of the DNA sequence. The pre-mRNA is then processed into mature RNA. This includes splicing out of the introns and the addition of a 5' cap and 3' poly A tail. Then the mRNA is ready for export to the cytosol and for translation. Now upstream of the gene, we have the promoter, which is composed of several elements. First, there's the TATA box, which is named for its nucleotide sequence and serves as an important binding site for general transcription factors. The core promoter includes the TATA box and part of the initiation sequence, which is right next to the start of the gene. The promoter also includes other proximal control elements, such as binding sites for specific transcription factors. The promoter is responsible for controlling when, where, and to what extent a particular gene is expressed. The basic idea behind a gene reporter is to use the promoter from a gene you're interested in, our gene X, to drive the expression of a reporter such as GFP or LAC-Z that will enable you to visualize or measure where and when that gene is expressed in an organism. Molecular cloning techniques can be used to copy the promoter from gene X into a plasmid that already contains a reporter gene. If this is now our plasmid, the promoter from gene X is inserted in front of the GFP gene and will now drive GFP expression. All of the transcription factors that normally bind to the promoter of gene X will also drive the expression of GFP. There are several important things to consider when designing or using a construct. For example, are all of the important control elements included? Is the reporter gene in frame? If constructed appropriately, gene reporters are powerful tools to understand gene expression in many different contexts. Let's circle back to the various ways gene expression is regulated. Where do all the techniques we've talked about go? The DNase assay can be used to test how accessible a particular gene is. EMSA can be used to determine if a protein is binding to a particular DNA sequence. This is often applied to transcription factors. qPCR and microarray tell you how much mRNA is present in a sample, relatively, which depends on transcription. qPCR can also be used to examine the expression of different splice variants. Western blot reveals the amount of protein in a sample, so it applies to translation. In addition, some antibodies are able to distinguish between different post-translational modifications, such as phosphorylation. IP is really concerned with the interactions between proteins, which is something that also happens after translation. Circling back again, a variation of IP is called CHIP or chromatin IP and can be used to investigate histone and DNA interactions. This has been a brief introduction to some important techniques in cell biology, and I hope it gives you the tools to reason and make predictions about experiments using these methods. To review, here's a triangle containing the 3 macromolecules that we've talked about. Pause the video for a moment to place each method near the appropriate one. Here's where I think they should go. I'm glad you joined me to talk about the techniques and methods to study gene regulation and expression. This has been Emily Jobe for BioCore 383 at the University of Wisconsin-Madison. Thanks for watching.